Hi, this is Elliot from EO Nutrition, and in today's video we will be examining the rationale behind using mega doses or extremely high doses of a nutrient called thiamine or vitamin B1. So this particular vitamin has been used in human clinical trials for a wide variety of different health conditions, and it has been shown to, in some people at least, produce major symptomatic improvement. Now, I have seen this on numerous occasions in my own clinical practice with the people that I work with. And again, I would like to try to explain that in this video, how that is working, because I do not think it is simply acting to replete nutritional status or address a deficiency. In fact, simply addressing a deficiency is not going to help. It's not going to explain the magnitude of effects that are seen when very high doses are used. In contrast, pharmacological doses, I think, are having an effect on cellular energy metabolism, restoring energy metabolism when it is being disrupted or inhibited by other factors. So, to quickly summarize, in very simple terms, there are lots of things which can disrupt how cells make energy. And when cells lack energy, they cannot work efficiently. So the idea behind, or the rationale behind, using megadoses of B1 is that it has a particular capacity to stimulate certain enzymes involved in energy metabolism and restore metabolic function in cells so that they can begin working as they previously did. And so this is the kind of thing that I see in clinical practice. This has been noted in the scientific research. We will go through that at the end of this video. We will also look at the potential mechanisms with regard to how this works. So hopefully by the end of the video, you will have a much better understanding as to why high doses or mega doses of thiamine are sometimes necessary to achieve certain effects which cannot be gotten through dietary levels. So first of all, to summarize, here are the key points. High dose thiamine has been used to improve the symptoms in a wide variety of different health conditions. I think one of the ways that this works is because thiamine-dependent enzymes can be selectively disrupted by toxins, by chronic oxidative stress, and inflammation. Now, what this does is it produces this same scenario, the same biochemical situation as having a deficiency. But this is not a dietary deficiency. This is a functional deficiency. Now, ordinary levels from the diet are not sufficient to overcome the inactivation of these enzymes. In fact, pharmacologic doses, so extremely high doses, can and do restore enzyme activity. So by saturating these enzymes with the cofactor, with thiamine, you can effectively restore normal energy metabolism. And because of this, someone does not necessarily need to be deficient in the dietary sense to benefit from this type of therapy. Now, briefly, we will need to go through the very basics of enzymes. This is just a quick run through, just so that you can understand the principles. And I go into this in a little bit more depth in the article, which is being linked in the show notes. But basically, what you have to understand, I've spoken about this in previous videos, is that nutrients, many times they act as cofactors for enzymes in the human body. That's how we use them. That's how they are necessary. And so enzymes are types of proteins which are responsible for speeding up biochemical reactions. Many enzymes, they help convert one thing into another thing, or they help build things or help break things down. That's all that you need to know. Now, enzymes, oftentimes, they need cofactors or coenzymes. It's basically the same thing. And the cofactor, when it is bound with the enzyme, it can effectively allow the enzyme to do its job. So in this context, it's converting A to B. Now, enzymes have an affinity for their specific cofactor, okay? So when the cofactor affinity is high, that means it binds very tightly with the cofactor, and this binding increases the rate of the enzyme function, or increases the rate of reaction. Now, on the other hand, if an enzyme has a lower coenzyme binding affinity, lower ability to bind with its cofactor, then we get a reduced enzymatic function or a reduced rate of reaction. So why is this important? Well, what you need to know is that there are a variety of genetic conditions which are inherited. And what this basically means, or something that is present in these conditions listed on the screen, is that the people with these genetic conditions, the way that they make proteins, the way that they make specific enzymes is slightly different to the average person. And that would depend on the gene that has the defect. So I'll give you an example. In a condition which is called B1 responsive maple syrup urine disease, these people have a defect in a gene which allows them to make a protein called branched chain keto acid dehydrogenase. It's a very long name, but essentially what you need to know is it's an enzyme and it helps us break down branched chain amino acids. And this enzyme ordinarily uses thiamine, uses thiamine as a cofactor. But in this genetic defect, the enzyme that they make has very low affinity for thiamine as a cofactor. And what that means is that the ordinary amounts that they get from the diet are not sufficient to drive the function of this enzyme. This means that these people can end up with severe metabolic dysfunction, and in some cases, they might die very early on. Now, the way that this particular variant has been treated medically is by using very high doses of thiamine. It's the same as the rest of these conditions and many other conditions. There's another condition called B1 responsive Leigh disease, and this involves a defect in the gene which encodes pyruvate dehydrogenase, another enzyme which uses thiamine. On the other hand, we have a condition called methylmalonic acidemia, and this again is a defect in encoding for an enzyme which uses B12. There's another one called B6 responsive homocystinuria. Now, all of these conditions, what they have in common is that they can be treated medically by giving very high doses of the nutrient cofactor specific for the enzyme, which is not being made correctly. How does this work? Well, in very simple terms, we spoke about how enzymes have affinity for their cofactor. And if someone is making enzymes with very low affinity, it means that it's only going to be able to bind a very small amount of the cofactor. It's going to be equivalent to this person being severely deficient in the nutrient, despite how much they get through the diet. Now, if we look at how nutrients can be used, by giving extremely high doses of the nutrients, in other words, by saturating the enzymes, we can overcome the lack of affinity. By giving mega doses of these nutrients, this lack of affinity can be overcome. We can bypass this defect by saturating cells or saturating enzymes with enough of the cofactor so that it can start working as it ordinarily should do. And so the large majority of people do not have these genetic conditions. However, we can use very similar principles and apply them to chronic diseases. Now, the reason for this is because it's not just genetics which can affect whether an enzyme runs or works as it should do or not. In fact, many of the enzymes which are involved in energy metabolism can be inhibited or activated via various factors. If we look at many of the enzymes, they are naturally inactivated or naturally inhibited as part of normal human physiology, right? We need to turn on enzymes at different times and turn them off based on metabolic demand. There are many other factors which can inhibit our enzymes, the enzymes which we use to generate energy in the cells. They can be inhibited, and when this goes on for long enough in a specific way, then this can become pathological. What this means is that this can contribute to disease processes. One specific type of enzyme is called alpha-ketoglutarate dehydrogenase. This requires multiple nutrients to function. One of those nutrients is thiamine. Thiamine is a cofactor for this enzyme complex. Now, this particular enzyme is what is referred to as a rate-limiting enzyme. It's a rate-limiting step in a cycle which is called the TCA cycle or the Krebs cycle. Now, what rate-limiting essentially means is that when this enzyme slows down in activity, every other step after it in energy metabolism is also going to be slowing down. Now, this can be beneficial in certain circumstances, but it can also be potentially detrimental when this goes on unnaturally. Now, it's important to note that this enzyme is uniquely sensitive to inactivation by oxidative stress. Now, this has been identified to be inactivated or inhibited in multiple chronic diseases, and it's been said by various researchers that under chronic conditions, when this enzyme is chronically inactivated, it likely contributes towards pathology. So heavy metals, excitotoxic chemicals, oxidized products of neurotransmitters, and neuroinflammatory products all have an inhibitory effect on this enzyme and likely contribute towards disrupted energy metabolism, and this is going to be found particularly in the brain. Now, what can lead to the production of excess amounts of these chemicals? Well, we see that there are lots of things that are involved in neuroexcitotoxicity, or lots of things which can have neuroinflammation as a component. So exposure to biotoxins, this can be mycotoxins, this can be other types of toxins produced by Lyme disease, for instance. This can modulate the immune system in the brain to activate the inflammatory response. We see EMF radiation, so electromagnetic frequencies, mobile phone radiation, Wi-Fi, etc. These can trigger the excitotoxic cascades, which increase peroxynitrite in the brain, increase nitric oxide, chronic infection, chemical toxins, heavy metals. Again, these things are ubiquitous in the environment. All of these things can contribute towards an inhibitory effect on this enzyme system. When there is chronic oxidative stress, when there is chronic inflammation, particularly neuroinflammation, we see that this enzyme is inhibited. Several of these chemicals also have an inhibitory effect on the pyruvate dehydrogenase complex, which once again is also dependent on thiamine as a cofactor. So two key energy producing enzymes or enzymes involved in energy metabolism, both requiring thiamine, are both inhibited by a variety of factors which are not related to a deficiency. And so how does this relate to the topic of this discussion? How does this relate to megadosing of thiamine? Well, a very interesting research paper came out in 2018. It was called Thiamine Preserves Mitochondrial Function in Rat Model of Traumatic Brain Injury, Preventing Inactivation of Alpha-ketoglutarate Dehydrogenase Complex. And so in this study, they showed that rats who had been exposed to traumatic brain injury, and what they found was that there was lots of oxidative stress. And what this did was it inactivated the alpha-ketoglutarate dehydrogenase complex. It reduced ATP synthesis, and there was lots of neuroinflammation as well. So basically, the damage caused by traumatic brain injury affected how the cells made energy. They made a lot less energy. Now, what they also did was they exposed another group of rats to a similar head trauma. But before that experiment, what they did was they pre-treated them. They gave them extremely high doses of thiamine pre-trauma. And what they found was that the rats that had very high doses of thiamine, they found that there was no inhibition of the alpha-ketoglutarate dehydrogenase enzyme. In fact, it remained exactly the same. They also found that mitochondrial function was protected. They did not find a reduction of ATP. In fact, the cells maintained energy production like they would have if they did not have a traumatic brain injury. They also found some indicators of reduced neuroinflammation. And so what they concluded was that megadoses of thiamine protected against the effects of this insult And this was outside the context of a deficiency. These rats were not deficient per se, but the trauma produced a situation which was very similar to deficiency. And by giving very high doses, they could overcome or they could bypass that inactivation. And so what do we see? We see that this is evidence of how multiple different factors, multiple different influences, whether it be any of those things at the top of this diagram, which can produce oxidative stress, which can produce chronic inflammation, these things will inhibit enzymes. The enzyme which is most susceptible to these effects is alpha-ketoglutarate dehydrogenase, and it turns out that that requires thiamine to work. So we use a similar principle, giving very high doses, saturating the enzyme with the nutrient. And what we find is that that can bypass this inactivation. So giving very high doses of thiamine beforehand prevented the inactivation of this enzyme and maintained energy metabolism as it was prior. Another study was also looking at traumatic brain injury. They were measuring levels of glutamate, which is involved in neuroexcitotoxicity. It's one of the excitatory neurotransmitters. And what they found was that the rats who had been exposed to traumatic brain injury, their brain had been injured, they had got massive amounts of glutamate in the brain, which was causing this excitotoxic cell death. Now, what they found was that very high doses of thiamine before trauma, pre-trauma, increased glutamate clearance in the brain and also protected the cells against cell death. Once again, this was outside of the context of deficiency. These rats were not deficient in thiamine before the experiment. So this is demonstrating that thiamine has a certain medicinal quality, protecting the cells by preventing the inactivation of enzymes caused by other factors. Another study was looking at neuroinflammation, which was caused by radiation exposure. Now, what they found was that high doses of thiamine before the exposure reduced inflammation and increased lifespan significantly. Another model of spinal cord injury showed greater clearance of nitric oxide, maintained levels of brain glutathione, which was highly protective in this condition. In oxidative stress, megadoses have been shown on numerous occasions to reduce markers of vascular endothelial oxidative stress. The authors of one study concluded the impairment of the alpha-ketoglutarate dehydrogenase enzyme plays a key role in glutamate-mediated neuroexcitotoxicity in neurons during traumatic brain injury. So in other words, it is the inactivation of this enzyme which is responsible for some of the cell death that occurs in injury. Now, here they go on to say, pharmacological activation, meaning very high doses of the nutrient cofactor using a similar principle as we've discussed previously. Pharmacological activation of this enzyme may thus be of neuroprotective potential. This has also been shown in relation to another one of the thiamine-dependent enzymes. We're looking at one called transketolase. Transketolase is involved in a biochemical pathway called the pentose phosphate pathway, and this is involved in recycling glutathione. There is research showing that by giving megadoses of thiamine, what this does is it increases the expression of this enzyme and it increases the activity of this enzyme. And this is theoretically one of the reasons why megadoses of thiamine can help to maintain glutathione levels even in the event of a severe stressor which is known to deplete glutathione. A similar principle also applies to an enzyme which we discussed previously, pyruvate dehydrogenase. This is using thiamine as a cofactor. Once again, there are multiple factors which have been shown to inhibit this enzyme. Now, this is ordinarily inhibited in a thiamine deficiency. However, these factors are separate to a deficiency. We're not talking about a deficiency. We're looking at oxidative stress, for instance, copper toxicity, cardiac ischemia. By saturating the enzyme with the cofactor, you can overcome that inhibition. You can bypass that inhibition And so high doses of thiamine have been shown to reactivate the enzyme. Now, if you want the details of these studies, once again, these are on my website. They are linked in the show notes. So just to summarize, we have this principle of enzyme inactivation. We have this principle of using high doses of a nutrient cofactor to stimulate enzyme activity, even when it's been inactivated. This has been demonstrated mechanistically, but in the human research, this is where it really shines. There was a doctor, late neurologist based in Italy. His name was Constantini. Now, he did studies using extremely high doses of thiamine hydrochloride, and he published his results in several journals. Just a brief overview of some of the effects or some of the results he got using megadoses of thiamine. He studied the effects of thiamine in fibromyalgia, in many of the symptoms, including the depression, including the fatigue, and he found abrupt and immediate improvement, and this was between 1,500 and 1,800 milligrams per day. In multiple sclerosis, or significant improvements in neurological symptoms at similarly high doses. In Parkinson's, one of the studies showed between 30 and 70% improvement, and some of the patients with a more mild form of the condition went into complete clinical remission as long as they remained on very high doses of B1. Other conditions studied include dystonia, Friedreich's ataxia, the fatigue associated with inflammatory bowel diseases, and essential tremors. Now the question is, were all of these people deficient? Were all of them dietarily deficient in the ordinary sense? And is this why they got such an immense benefit from taking thiamine in such high doses? Well, I would say that that's probably not the case. Now, to quote Constantini here, he said in one of his research papers, "From this clinical observation, it is reasonable to infer that a thiamine deficiency due to enzymatic abnormalities could cause a selective neuronal damage in the centers that are typically affected by this disease." I think he was talking about Parkinson in that particular study. Now, note that he said a deficiency due to enzymatic abnormalities. So these people probably getting enough in the diet. Now, this is very similar to what I've been speaking about, this concept of a functional deficiency, this concept of someone having the enzymes involved in energy metabolism inhibited. They're inactivated by other things. And this produces a very similar situation to being dietarily deficient. But the amounts that you can get from the diet are not sufficient to overcome this inactivation. Here is another quote. "We may suppose that symptoms decrease when the energetic metabolism and other thiamine-dependent processes return to physiologic levels." So basically saying that by giving very high doses, you are returning thiamine-dependent processes back to normal. You are restoring whatever deficit was there. However, they say our aim was not to correct a systemic deficit of thiamine, but rather to increase the activity of enzymes involved in cell production of energy in selective brain regions. Here we go. So this is exactly the kind of concept I'm trying to explain, is that we're not looking at addressing a deficiency, but rather we are correcting the activity or stimulating the activity of specific enzymes, thiamine-dependent enzymes, this being pyruvate dehydrogenase, this being potentially transketolase, but this certainly, most certainly being alpha-ketoglutarate dehydrogenase. By giving very high doses, we can restore normal energy metabolism even when those enzymes have been inactivated by the factors which we've previously discussed. This means oxidative stress, this means toxins, this means inflammation. Okay? Some other reasons why thiamine might also be achieving a benefit at such high doses is that when it's given, it exhibits anti-stress properties. Here are some of the things which have been found in multiple research studies on a form of thiamine called benfotiamine, showing that this nutrient can be highly anti-inflammatory. It can reduce markers of oxidative stress in the cells and act in an antioxidant fashion. It's important to note that in some of these studies done by Constantini, benefits were not seen at lower doses. In fact, in fibromyalgia, two of the subjects only saw benefit once they reached 1,500 milligram, and they only saw significant benefit when they reached 1,800 milligram. He described this as an all or nothing effect, in that doses anywhere below that had basically no symptomatic improvement, whereas only when they reached a certain threshold that they saw great improvements. To summarize, high doses of thiamine can be used to overcome metabolic blocks. These metabolic blocks can be caused by a variety of different problems which are not related to a deficiency in the first place. This can be traumatic brain injury, this can be neuroinflammation, this can be chronic oxidative stress, this can be heavy metal poisoning, this could be any number of different things, okay? And these physiological conditions, the neuroinflammatory processes, the chemical toxins, the oxidative stress, these are present in a wide variety of different health conditions. This can be chronic fatigue syndrome, it can be ADHD, it could be fibromyalgia, it could be any number of different conditions feature some degree of these kind of processes taking place. And it's theoretically possible that one of the reasons why thiamine can benefit so many people in when it's used in high doses, and this is something that I can corroborate clinically, that I see clinically, and I want to understand. I think one of the reasons why this works is because it's having a stimulating effects on enzymes which have been blocked. And thiamine, when it's used to sufficiently saturate cells, you can help to overcome that block. You can help to restore normality, or you can help to normalize how cells can make energy and allow them to return to a place where they can function as they need to function. I've seen people from all walks of life benefit from this particular nutrient when given in a high dose. And I don't believe that they are classically deficient or even insufficient in the dietary sense to benefit from this. In fact, these people oftentimes, they follow a really clean diet. They have done lots of work on their digestion. They've done lots of work on their gut health. They've done lots of work on trying to optimize their nutrition. And it's only when we get up to the 500 times RDA or 1,000 times the RDA that they see major improvements. And this is exactly what Dr. Constantini demonstrated in his research studies. What it seems to be doing is it's either acting on some kind of a genetic level to increase expression or production of enzymes of energy metabolism, or it's simply saturating enzymes that have been inactivated and restoring some kind of function. If you like this video and you found it helpful, please like and subscribe. You can find me on Facebook as EO Nutrition. Subscribe to my YouTube page. You can find my website at www.eonutrition. And I will be making more videos like this in the future. You can see the full reference article on my website, which is linked in the show description. So until then, I'll see you next time.